cristae. So if I'm looking at this, this is the mitochondrial inner membrane here. So this is mitochondrial inner membrane. This is outer membrane. These are cristae. And my ATP synthase is somewhere in here. So I've got this drawing now. So I have a feeling for like, this is what it's going to look like. So I'm starting this. And if you use the mitochondria for a lot of things, you can answer a lot of questions. So then I'm going to draw this membrane. If I take this out of here, and I say, okay, now I'm going to take this membrane out of here, inside membrane. I'm going to have the intermembrane space. I'm going to take this out of here, and I'm going to draw, again, a kind of a plasma membrane here. I'm going to just draw this line here. So I'm looking at a membrane. You're looking at a lot of membranes. And so I'm going to make my little amphipathic structures in here, like this. And I'll just put them over here at the other end so I can see where I'm at. And this kind of configures my brain to think about this as a membrane. Then I can start putting in the 5 complexes that go with the chemiosmosis and electron transport system. So I've got complex 1 here, and let's say we'll just for time's sake, we'll draw it like this. We'll just make it like this. Then we'll make complex 2 here. Then we'll make complex 3 here. I might have to make this a little bigger.
Excuse me, I missed what you have said. So you are drawing the
inner membrane, yeah,
okay.
inner membrane. So let me just make this a little expanded here. So this is going to be complex 1, complex 2, complex 3, and then I'm going to put complex 4 here. And then lastly, and we got, wait a minute, we got one more. We got complex 5 over here. There's all these protein parts of it. They're very complicated. And then lastly, I have ATP synthase. And I think there's a really cool structure. There's about 12 subunits on it that are all similar. Then it has this is all cylinders I'm drawing, rotating arm, and then this structure here, like this. And then it has this connecting factor here, like this, and a connecting factor here, like this. And this is, and we're going to just outline it. These are proteins. This is a protein. They're all made out of proteins. And so this is ATP synthase here. Big enzyme complex. And this is all proteins here made out of it. This looks like some kind of rotating thing, and it is. So now I've got my membrane, and I've got the inside membrane, I've got my complexes. Now, how am I going to answer this question? Well, I'll tell you how. We're going to go like this. You've got NADH here. And NADH is going to give up electrons and become NAD. Now that I drew this, I know what I'm going to do. These electrons are going to raise this complex to a high energy level. It's going to be excited. And there's 2 things here. There's coenzyme Q, and then there's cytochrome C. Now, the whole point of this thing is to get electrons into this complex. So electrons are going to be in here, and the proton is going to be pumped out. So I'm going to pump protons out as NAD comes in. And then this electron is going to be passed to coenzyme Q. And it's also going to be passed to this little one here. And then I'm going to pass it to this, and then I'm going to generate more hydrogen ions, because FAD is going to come in, FADH, and that's going to convert to FAD. And then I'm going to pump more hydrogen ions into this area. and then so on and so forth. So what am I doing here? I'm creating a hydrogen gradient, right? I'm creating a lot of hydrogen. There's a huge hydrogen gradient. So that's what's happening here. And by drawing this, I now understand what's going on here. And this question, so what are they asking me? Well, I'm going to ATP, ADP. How are they going to do that? So at the end here, you've got this huge hydrogen gradient. This is going to cause, and this is really cool, this thing to rotate because electrons are going to pass through here. This is a channel into this thing here and rotate this and rotate this. This is F0, this is F1. This doesn't move, but this will move. And if you looked at a cross section of this, let's say I took a cross section out and I looked at this, the shape of this, I would see the parts that make this rotating thing up. And every time ADP comes in here and converts to ATP, it changes the conformational shape of this moving protein motor. And I end up making phosphate coming in here, and I get a lot of ATP. So this whole thing here is the electron transport chain, and this is chemiosmosis. Now, you can watch a video of this and make your sketch with it, and you will understand it 1,000% more if you didn't do this. So now I can answer this question. I've got this ATP synthase. It's using the flow of protons back into the matrix to catalyze ATP from ADP and inorganic phosphate. Question, a patient taking DMP as a weight loss aid would experience which of the following symptoms? A, a decrease in oxygen consumption due to the ability, the electron transport system to function properly? No, because it wouldn't be functioning properly. B, an increase in ATP production? Would that happen? It's causing more hydrogens, think about it, and the inability, well, was it leading to surplus of excess energy? Maybe not. C, a significant increase in body temperature? You don't think so? Okay. And D, a decrease in glucose catabolism to compensate for overproduction of ATP. Okay. So think about this. You drew this. Now, you're getting all of this extra hydrogen. Can it actually use that? No. So what it's going to do is, it's going to dissipate it as heat. It's not going to go into the system. And so it's not part of it. It's thrown the homeostasis off. So it's going to dissipate heat. So the actual answer is, you're going to get an increase in body temperature. So I answered that by doing this. I might not intuitively think that's going to happen, but when I see all of these being generated up here, and I think about it, and I've done this ahead of time, then I sort of understand that that's actually what's going to happen. So the answer is the energy from electrons passing through the electron transport chain is conserved in the proton gradient. When the gradient is dissipated by DMP, which is what it does, it's going to dissipate. The energy is released as heat because it's not going to become part of that system. And that's going to change the actual temperature of the person's body. Because you might put more in there, but there's no way it can be processed, or there's no way it can go because it's not part of that system. This is making extra that wouldn't be. There's no NAD there, there's no FAD there, it's not transferring any electrons or any hydrogen. So this answers your question. So if this is a hard thing to think about, so if I had trouble with this area of biology, I would draw this multiple times. I would watch animation videos of it, and then I would say, how can I draw this again so I understand this concept? Because many times you have to redraw concepts to actually understand them. So that's kind of a way to think about that. Now I had a fourth one, I'm not going to do it with you, but it's very easy to do when you want to answer something about the loop of Henle and about the kidney. It's the same idea. You draw the big picture concept, and then you think about the picture concept as you're asking the question. So we get rid of this, and we say, well, I'm going to do a question about the kidney. They may ask me a lot of questions about the kidney. So I'm gonna draw a kidney, so I can have a rough idea. And again, this is just doing it very quickly, so you just have a rough idea. And I'm gonna draw the pyramids in the kidney. And then I'm gonna draw this, which is gonna be my ureter coming out from the calluses of the kidney that are leading into it. And I'm gonna get asked a question about a nephron. So the nephrons are all in here. So I kind of need that picture to make the understanding of the nephrons go through. So, what are they gonna say about this? Okay, so I'm gonna just draw a quick nephron, so I know. Here's my Bowman's capsule, the glomerulus, and here I'm gonna draw, this is in this space here, this nephrons, so I know they're here. And I'm gonna draw this kind of convoluted tube, and I'm gonna go back up, I'm gonna draw another one and go to the collecting duct, which again is a cylinder. And I'm gonna follow this through, and this ascending limb is much fatter, and I'm gonna go to the loop of Henle, and then it's gonna get skinny, and then I'm gonna go back to the proximal convoluted tube. So then I've drawn a nephron. I've drawn a nephron, and I can answer my question, because in the nephron, this is Bowman's capsule. This is the glomerulus in here, which is all the capillaries. And I can just give myself a feeling they're winding around this tubule system here, like this. And this is my collecting duct. And I'll just speed it up a little for you. At the end of here, I'm trying to make urine. So my whole goal of this, I must say my goal, and put it here, is to make urine and adjust the blood, remove the toxins, and clean it. So osmolality, all those other things. So how am I going to answer this question? The distal convoluted tubule, which is this one right here, is fine-tuning water and ion reabsorption under hormonal control. Aldosterone increases sodium reabsorption, while antidiuretic hormone increases the permeability of the collecting duct to water, allowing for concentrated urine to form when the body is dehydrated. So I've got two hormones there, and what I'll do is I'll sketch them. I know that on the kidney is an adrenal gland.
Caroline, you have a question.
Oh, sorry, I have a question. Where in our body is the DCP? The DCP, the distal.
The convoluted tubule?
Yes.
That is on this structure called the nephron. So like, this is the Bowman's capsule, this is the proximal convoluted tubule, this is the distal convoluted tubule, this is the collecting duct. This million of these whole things are in the kidney. The kidney is made of all of this tubule system. It's made of the whole thing. There's a million of these sitting in the kidney. These are the functional unit of the kidney, called the nephron. So they are microscopic, you can't see them, but this is where urine is made. So blood is actually filtered through this whole thing, and urine is produced so that your blood can be cleaned and filtered. So like when your blood isn't cleaned and filtered, then the blood becomes uremic, it gets filled with nitrogen compounds and toxic, and it can be deadly. So kidney failure is when nephrons do not function well in the kidney anymore. But this tubule system makes up the whole kidney. That is the kidney.
Okay, and then a follow-up question, sorry, sir.
No, it's not.
If to create the kidney failure, you said that the nephron, the nephron has to let it stop or something?
Well, what happens is many things can cause the kidney to fail. It can become fibrotic. It can be over, too much pressure in it. Different drugs can cause it to not function well. Certain things can just, diabetes can enhance its deterioration. So when these do no longer can do that kind of normal filtering of the blood, then whatever the reason causes, then the kidneys fail, yes. And, you can survive with one kidney, but if you have a good kidney and was just damaged, then you're all right. But if you have a whole body that's been affected and your kidneys are affected, then both of them are typically affected. And then you need to go on dialysis. Dialysis is where they try to do the same thing the nephron does. They take the blood out of your body, they try to adjust solute, and they try and give your blood back to you as clean as possible. So that's how that, so yeah, this is the really critical functional part of the kidney. So if you answer a question about it, at the end here, you've got aldosterone, which is a hormone, working on this, and you've got ADH working on this. And they're both adjusting two different things. One is adjusting water, one is adjusting sodium, because sodium and chloride accumulate here. So they're trying to adjust this before it comes urine. You have too much sodium in your blood, you have too much chloride ion in your blood, it's adjusting it back and forth. So aldosterone sits inside of the adrenal gland, which is on top of your kidney, and ADH comes from the posterior pituitary gland, which is in your brain. So you can make a picture here like this and say, okay, this is the brain, here's the pituitary gland right here, make it a little bigger, and this ADH is coming from that posterior pituitary gland. So that's going to adjust the urine. That hormonal stuff is going to adjust the urine at the end. So your question is, a patient has a genetic defect that makes the ascending loop of Henle impermeable to sodium and chloride ions. Which of the following physiological consequences would most likely occur as a result of this defect? So look at the possibilities. One, a decreased water reabsorption in the descending limb, leading to production of very dilute urine. B, increased reabsorption of glucose and amino acid in the proximal convoluted tubule. That's the one closest to the nephron. C, excessive sodium and chloride reabsorption in the collecting duct, leading to hypernatremia. And D, increased osmolarity of the medullary interstitium, enhancing the effect of ADH. So think about that. So you're concentrating the urine, back here you drew this, you're adjusting, antidiuretic hormone is working with aldosterone to adjust the urine. And so you made this picture, and I mean, I'm doing this fast, but let me get to the answer. So the correct choice is A, the ascending loop, normally pumping sodium and chloride into the medullary interstitium, creating the osmotic gradients that drive the reabsorption of the descending limb and collecting duct. If this active transport is disrupted, the osmotic gradient can't be maintained and the water reabsorption in the descending limb decreases. So what you're going to have is a nephron that's losing its ability to concentrate urine, and you're going to have very dilute urine as a result of it. So my point is, if you make this diagram and you illustrate this, you can answer lots of questions about the kidney. If you know that this is going to be a topic that's going to be on your exam, then you say, I'm going to make this very important illustration. This and this together can answer a lot of questions. You just have to make it a little better than I did, but it's not hard to sketch out, because look at how we sketched this. This is nothing. You could make 3 circles here, like this, and make your kidney, like this. And you could use a curved cylinder here and make your ureter like that. And what is this? This is basically the Bowman's capsule, and I'm making a line, and I'm making a loop, and this is doodling and sketching. And then I make a second one so that I can make the thin loop here that goes down to the loop of Henle, make the thick one here that goes back to the distal convoluted tubule, and then the collecting duct. And I made it that easy. And then I can diagram my idea out, and I can figure out how to finish this question or how to answer another question. So this is kind of, the way I think would be a better way to study for things than just searching internet. And of course, if you don't want to use it to study, you can also use it for biological discovery. So you can use it to form hypotheses, you can use it to make ideas, and all of these are old sketches that people made before they came up with really great ideas. This is the stem cell diagram over here, which had to do embryologically with the way cells branched out over time and became different parts of the embryo. That's like 100 and some years old. And this is, again, a original sketch of mitosis, where chromosomes exist in the cell, what they do, how they divide. This is an old Chinese drawing, a zoological drawing of a zebra. It's a sketch, but it's also a name. Yeah, it's really, I think it's Arabic maybe.
No,
Persian. Oh, it's Persian. Okay, I get a Persian one. Okay.
yeah, I can read that.
You can read that?
Yes.
Okay, sorry. And this is out of Linnaeus's journal of his botany on plants. This is, people were usually kind of documenting about the venation and the patterning of butterfly wings to understand the evolution of them and the relationships between them. And this is somebody's from 1800s, small sketch of a squid while they were on a boat navigating. So you can use all of this to think about your biology and to change the way you think about the way you do your science in a lab and how you might approach a question. And certainly it helps in computational circumstances because it gives you some nice contrast to be able to keep a sketchbook, be able to write things down, be able to actually reason them out. So I'm kind of like going to go through this quickly, but you know, it's a much different way to think about biology because there's flow involved in it, and flow is a real important way to generate your creativity because it isn't a pipeline of next this and next that and next this. It's just things that happen here and there that influence the way you think about a subject or an idea. And that is where creativity comes from and good questions get asked. So if you're interested, this guy Mihaly, this says Mihaly, I can't pronounce his name. He wrote this book called Creativity and Flow. It's really worth reading. He talks about a lot of scientists and artists and how they practice this and how it's part of their life. And so they feel much more fulfilled and creative when they do it. So I'm just adding that to any of you who are interested in doing research. And it's really two paradigms here where you can fuse your flow with your pipeline thinking, which is reductionist, digital, and extractive. And the thing is, it's nice to have that flow in there because it's a very good feeling. These are just some journals by Leonardo here, and you can interpret ideas and develop thoughts and concepts about them, and look at nature and study it so that you can get a feeling for what these things look like. Here's just some more examples for you to look at, some people's notebooks, this one on fungi, this one on protozoa. So I wanted to just share this with you, and we looked at the ribbon model and the original drawing of chromosomes from under the microscope. And this is a methodology and a conceptual tool, as well as a tool for studying. So my recommendation is you get a sketchbook and you just get a pencil and put yourself a half an hour a day, and let yourself draw out your ideas from your from your workbook, from your courses, from the exams you're going to take, from questions you can't answer, from concepts that maybe you didn't get in class. And maybe you need to just sit down and put a big focus into it, and you'll find out over time your sketchbook will get better and better, your ability will get more enhanced, and you'll have more use for it. I know in the digital age it's hard to put those things down, but the fact is this is like a real lifesaver when you want to have some peace of mind and you want to think deeply about a subject and you want to give it your focus. And you want to pass a test, especially if you don't get the concept. The thing I would do is I'd look for a good animation, I'd read the text I'm supposed to have, and then I would make my sketches. And that's how, if I couldn't get it in class, I'd go home and I'd work through it that way. And I think you'll find that it'll really answer a lot more than just kind of trying to memorize things and trying to get them into your head. You know, not everybody is going to give you a direct line to what the question is. Not everyone's going to help you pass things. You're going to have to figure them out yourself, and this is kind of a self-generating tool, so it's really important to have it. I think that's it. So I finished. I did have a card for everybody that we wanted you to take with you.